Navin, shall we start then?
Yeah, let us start. Yeah.
Okay, so let me welcome everybody, particularly Professor Rao, the guest speaker today. And before I introduce Professor Rao, I'll briefly say two things about the audience to Professor Rao. We are a very diverse group, faculty from science, engineering, and humanities and social sciences. And of course, all of us are keen to understand how important the particular work which won the prize is. So we would be very keen audience, of course. So let me introduce Professor D. Narasimha Rao. He is currently a professor of biochemistry at the Indian Institute of Science, Bangalore. Uh, and he has been a faculty there for about 31 years from 1989, and he also is simultaneously the chair of the division of biological sciences for about 15 years, almost half his career there. He has been also a chair. Now, chairmanship of of a division in IIC is a very uh, it has a lot of responsibilities. There are 8 separate departments or centers under that particular division, and one of them, very well known, of course, including biochemistry, is molecular biophysics, which was started by Professor G. N. Ramachandran, the very famous scientist from India, who also worked on molecular biophysics. There are around 70 faculty in this division, around 400 PhD scholars. So you can imagine how large that is. And Professor Rao has been the chair of that for about 15 years now. Professor Rao's academic career started in Delhi, although he hails from Mysuru. He did BSc in Delhi University, an MSc from GB Pant University, Nainital, and then PhD from IAS in 1982. From in both GB Pant University and in IASC, he is an outstanding alumnus, recognized as an outstanding alumnus. Professor Rao left in 1982 and then went to 3 major institutes in the world to do postdoctoral fellowship. One in NIH USA, the other in University of Basel, and the third in University of Cambridge. And as you know, it was Crick and Watson working in Cambridge, collaborating with Imperial College, who produced the structural structure of DNA, an outstanding work. Uh, Professor Rao has been recognized and given many awards. He is a fellow of all the 3 Indian science academies, and he is honorary professor of IASC for a term of 5 years, starting from 2020. He was earlier an honorary professor of CASR, and he has won many other major awards. He has had supervised numerous students, has major publications. So I wouldn't go into those details. I'll briefly say a few things about the research that he does, which he articulated very well in his webpage. So layman also can understand. There are 3 strands there. The group tries to understand how certain enzymes recognize, cleave, and modify DNA. And the group tries to understand precise interactions of a particular protein with DNA. And very topical, it also tries to understand protein-protein interactions of a particular human pathogen called Haemophilus influenza. Uh, and 2 things very important about Professor Rao. He is on the board of Resonance, which indicates the keenness he has on science popularization. And we have heard that he is one of the most student-centric professors in IISc, giving a lot of importance to students. So with this, it's a pleasure, sir, to have you with us to give this talk. And I
All right.
invite you to talk on your topic titled Genome Editing and the Breakthrough Work That Could Change the World.
All right. Thank you very much, Dr. Kurian Isaac. It is very nice. And thanks to Jayanti and all of you for having me to here this afternoon to give a talk on the Nobel lecture in chemistry that was just awarded a few days back. I will. I've realized that there are no biologists there, and all of you are more very brilliant physicists, chemists, and mathematicians there. So I will try to make it simple. And although I might have a few slides which are a bit giving you some details, I will try and explain them in a more simpler way so that you get the gist of what the whole Nobel Prize is all about. I will just put this full screen. Is that working?
Yeah, it is become full screen. Only the matter hasn't come yet. No,
All
now
right. Okay. All right.
now it has
Yeah.
come.
All right.
Now it has come up.
All right. The 2020 Chemistry Nobel Prize was given to two women scientists, absolutely brilliant scientists, Emanuela Charpentier from the Max Planck Institute for Infection Biology in Berlin, and Jennifer Doudna from the Department of Chemistry and Biomolecular Sciences at the University of California in Berkeley in the United States. And the award says very clearly for the development of a method for genome editing. And I emphasize this this citation, a method for genome editing, because there are earlier very nice methods that people have described for genome editing, but it is this one, the particular one that Emanuela Charpentier and Jennifer Doudna have actually popularized this method of genome editing, and I'll tell you why it's become so popular and so famous. Uh, Both of them in 2011 met at a meeting in Puerto Rico and published the Nobel Prize winning work in science in 2012. So, actually, completely independently, that these two scientists hit upon the same problem and then decided to collaborate, and this whole editing of genome became a very fashionable thing and more popular and very easy to do. And I'll tell you why it is easy to do. But before I go into the work of Emanuela Charpentier and Jennifer Doudna, I thought I'll just spend a few minutes telling you about the Nobel Prize in Chemistry. And I think that is what we need to appreciate, that the Nobel Prize in Chemistry, there are about 590 or 91 Nobel Prizes in chemistry that have been awarded since 1901. 110 out of 590 are the chemistry prizes. They were not awarded in certain years because of the World War One and World War Two. And the most number of Nobel Prizes in chemistry has been awarded to scientists working in biochemistry, which is a sore point among many chemists because the more recent ones are all going to people who are doing biochemistry and studied biochemistry. The first Nobel Prize in chemistry was awarded in 1901 when the prizes were actually instituted, and that goes to Van 't Hoff for the discovery of the laws of chemical dynamics and osmotic pressure in solutions. Frederick Sanker, whose picture is shown on the right side, extreme right here, is the only Nobel laureate who received two Nobel Prizes in chemistry in 1958 and 1980 for describing the methods of protein sequencing and nucleic acid sequencing. Marie Curie got two prizes twice, once in physics and one in chemistry. And Linus Pauling is the other person who got the chemistry prize in 1954 and later on the Nobel Peace Prize in 1962. These are the only two people, these are the people who have actually got the prizes twice. The youngest chemistry Nobel laureate is Frederick Joly, 35 years old. And the Curie family, also referred to as the Nobel family, in short, has received 5 Nobel Prizes. Marie Curie, Pierre Curie, husband, then Irene Curie, Frederick Joly, and then Eve Curie in 1965 as a part of the UNESCO Nobel Peace Prize. So the prices, as you see now, and as has been the thing, that very few women have actually got the prize in any of these subjects that in any of the areas that the Nobel Prize gives. More so in chemistry, although there are 110 or 113 prizes that have been awarded, there are only 7 women who have got the prize. And I thought I will just show you the picture of them. And this is the four, which is in one slide. These are the Curies, the Marie Curie on the extreme left. Then in the third picture that you see here is actually Dorothy Hodgkin, uh, who got the prize for X-ray crystallography studies. And the extreme right is Ada Yonath, who got sometime back, a few years back, who shared the prize with Venki Ramakrishnan for the structure of the ribosome. Absolutely fantastic piece of work that was recognized. And Ada Yonath, Venki Ramakrishnan, and Tom Stites got the prize in chemistry for their discovery. 2 years back, Francis Arnold from the California Institute of Technology got the prize in chemistry for directed designer enzymes, directed evolution. And last 2 years back, I've been speaking about this Nobel Prize in chemistry at a number of places. And this year, as you see at the bottom here, is Emanuela Charpentier on the left and Jennifer Doudna on the right, who got the chemistry prize this year for their development of a method of genome editing. Before I tell you more about the prize and the work, I'll give you a bit of history. And I think this is very important because, as I said in my first couple of sentences, this was a method of genome editing. And for that, we go back almost 60 years ago, where a Swiss biochemist, Werner Arber, at the University of Geneva, and his laboratory assistant, Daisy Duzua, actually provided the first evidence for the existence of molecular scissors that could cut DNA. And he showed that E. coli, which is the organism that all of us use, which is the laboratory animal, our bacterial system now, have an enzymatic immune system that recognizes and destroys foreign DNA, but in turn modifies its own DNA to prevent self-destruction. Very, very simple experiments in microbiology, just by plating, bacterial virus, which we call as bacteriophage, onto bacteria and seeing if the phages survive or don't survive. Very nice set of experiments, 4-5 nice papers by Daisy Duzua and Werner Arber actually gave us this whole concept of something called as a restriction modification phenomenon. And that is what is shown here. Daisy Duzua's experiments clearly showed, somehow this arrow on my thing is not working. It's showing here, all right. That this is the E. coli, the host bacterial chromosome, which I show here, which we call as a host chromosome. There are small organisms called bacterial viruses or bacteriophage, which cannot exist by themselves, but they have to come inside the cell, bacteria, to survive, to multiply, and to replicate and things like that. So the phage has its own DNA. The chromosome or the E. coli, the host chromosome, the host also has its own DNA. But the DNA here is actually quite indistinguishable from the DNA that the phage has. And therefore, you have in the host DNA, the DNA is modified, is methylated at certain residues like adenine and cytosine, which are the bases, which are the common bases that are present among the GATC. You have the adenines and guanines, which are adenines and cytosines, which are methylated. This methylation, by putting a methyl group here, is done by the host, by the bacteria itself, by the E. coli, and by an enzyme called a modification enzyme or a methyl transferase, which transfers a methyl. So once the bacteria, the host DNA is modified, the restriction enzyme, the enzymes that cuts DNA, is made, and when the phage DNA actually enters the cell, which enters the bacterium, that DNA is not methylated at all, and therefore the unmethylated DNA of the foreign DNA, which is injected, is recognized by the host restriction enzyme, which is a molecular scissors. Is an enzyme that cuts DNA, and the phage DNA is chopped off into a lot of pieces, and the phage does not survive. The same restriction enzyme, which chops off the phage DNA, cannot cut its own DNA because the DNA is exactly like the phage DNA, simply because the DNA is modified or methylated. And therefore, these deductions came from Werner Arbor and Desirazu's experiments, very simple experiments, which formed the whole phenomena, which formed the basis of a whole phenomena called the RM phenomena or the restriction modification phenomena. Just by looking at this whole phenomena, you can simply realize that this is a survival defense mechanism as far as the bacterium is concerned. The bacteria or the bacterium doesn't like foreign DNA, and therefore it chops it off. And therefore, this is a distinction between self DNA and non self DNA. And the way this distinction is done is simply because of the methyl group on the host DNA, which is not there in the phage DNA. And just by simple plate assays, by plating phages on bacterium with different backgrounds, Desirazu and Werner Arbor came up with this whole thing, phenomena. And the major role for this RM system is in evolution rather than in vital cellular processes. Now, this is a innate immune system that is inherently present in every bacterium. Every bacteria that you study, bacterium you study, it has one R gene and an M gene, a restriction gene and a modification gene. You cannot have the restriction gene alone because it will kill the DNA. You must have the M next to the R because the M is made first, it modifies the DNA, and then the R is now able to act on foreign DNA like the phage DNA and then chop it off into pieces. Towards the end of my talk, I'm going to tell you that phages have become very clever. They have now, they have over the years during evolution, as we talk about, they have developed system called the anti-restriction systems. And if I have time, I'll talk to you about a little bit about the anti-restriction system. But this battle or the, what is called as a war between the bacterium and phage is continuously going on. And as we speak, either the phages are superior or the bacterium is superior because one has overcome the other's defense mechanisms. So this is a very primitive immune system. Primitive because you are talking about bacteria, the first creatures on during in the evolutionary tree. And 60 years back, this is actually in the 1960, 1959, the papers came out from the University of Geneva from Desirazu and Werner Arbor's lab. Once these papers came out, everybody who was trying to do molecular biology kind of work or genetic engineering work were actually stunned to find that we could isolate now enzymes which cut DNA. That is the R that I was talking about, the restriction gene. So they all went back to their favorite organisms, grew fermenter, liters and liters of the bug, and isolated these enzymes. The biochemists thrived in this era where the enzymes were purified, and therefore now we could use these to manipulate DNA. You could cut DNA by using of restriction enzymes. And that is where the implication of Werner Arbor's work is. The potential to do fantastic things later on. And that is the basis of the Nobel Prize that was given in 1978 to Werner Arber for his outstanding discovery on restriction modification enzymes. He shared the Nobel Prize with two others from Johns Hopkins University, two scientists, Dan Nathans and Hamilton Smith, who did a lot of biochemistry and actually purified these enzymes and showed and applied it to problems of molecular genetics that you could cut DNA and cut DNA wherever they want by using a battery of these enzymes. Every bacterium has at least one restriction enzyme. There are some bugs which have more than one. A bug that I work on in my laboratory, Helicobacter pylori, which causes ulcers, gastritis, and is a carcinogen, is the only bacterial or carcinogen, has 44 restriction modification systems. So it's always a very interesting model system to study protein DNA interactions. And therefore, once you purify and once biochemists get into this whole game, the idea is to find out how these enzymes act on DNA. How do they recognize the sequence? And these are all sequence specific enzymes. They recognize a particular sequence. I'll show you a slide later on. And there's one thing that I picked up from the internet where Dan Nathan's daughter, she actually wrote, "My father has discovered a servant who serves as a pair of scissors. If the foreign king invades a bacterium, this servant can cut him in small fragments, but does not do any harm to his own king. Clever people use the servant with the scissors to find out the secrets of the kings. For this reason, my father received the Nobel Prize for the discovery of the servant with the scissors." So this is just Dan Nathan's daughter when he was awarded the Nobel Prize. So to just summarize the absolutely fantastic work that Werner Arber did is shown here, that you have a phage DNA which tries to inject this DNA into the bacterium, but the bacterium chromosome, the bacterial chromosome, produces two enzymes called the endonuclease, the restriction endonuclease, and a modification methylase. The modification methylase would methylate the DNA, the host DNA, and therefore everything is methylated before the phage is actually injected. And the restriction enzyme is made, and at the minute the phage DNA enters, these molecular scissors will chop off the phage DNA, and then you don't have phage multiplying at all. Now you must remember, phages, as I said, also have methods to counteract this, which is the anti-restriction system, which has now become a very hot area in biology to study how phages survive. You must remember also that the number of phages in our planet is a billion-fold higher than the bacteria that we have. We have 100 trillion bacteria, and we have at least 100-fold or 1,000-fold more phages that you have than the bacteria. So the phages, being the smallest living organisms, are many more than any other species that you have on this planet. One quick thing about restriction enzymes, just to tell you that these enzymes naturally occur in bacteria. These bacteria use these enzymes to protect themselves from viruses, which we call as bacteriophage, which invade them. And restriction enzymes cut up the DNA that the virus injects into the bacteria, so the virus cannot be reproduced. So that's the whole phenomena of restriction modification. As far as the bacterium is concerned, it is a defense mechanism. But as far as men are concerned, humans are concerned, this has opened up this whole revolution in biotechnology. All your genetic engineering, all the recombinant DNA technology is because of the discovery of these enzymes that you're able to cut. And by another enzyme which actually joins DNA molecules called the DNA ligase, you can summarize recombinant DNA or genetic engineering in one thing as a cut and paste. One cuts it and the other enzyme pastes it. In other words, you're creating a phosphodiester bond, and a phosphodiester bond is what you're actually cleaving by restriction enzymes, which is shown here. These enzymes, as I said, are very sequence specific. Here's an example of EcoR1. This is isolated from Escherichia coli, a serotype called R1. There's R2, there's R3, and things like that. But this is just an example that this enzyme derived from bacteria recognizes the sequence. Wherever in the double-stranded DNA, you have a sequence called GAATTC, the enzyme would recognize. The enzymes, like restriction enzymes, most restriction enzymes, recognize palindromic sequences. So that in one strand, you have GAATTC, and in the other strand, complementary to that, you have GAATTC in the reverse direction. Remember, the polarity is 5' to 3'. The other strand is 5' to 3'. So this is a palindromic sequence. This is just like the word Malayalam. If you read it in the forward direction or the reverse direction, that's a palindrome. So restriction enzymes, many of them, not all of them, recognize palindromic sequences. And once the enzyme recognizes the sequence, that is when it binds to the sequence, and the binding is all hydrogen bond or by hydrogen bonding, it will now cut the DNA between G and A and G and A here. This is the specificity of the enzyme called EcoR1. So when you take a DNA which has a recognition sequence like this, and you treat it with EcoR1, the phosphodiester bonds are broken, and you'll get these two different fragments. If you now take these two different fragments and add the enzyme called ligase, DNA ligase, in presence of ATP, because energy is required, then this will join together. The whole recombinant DNA technology is actually cutting and pasting, whether it is from bacterial DNA, a fungal DNA, a plant DNA, a mammalian DNA, or a human DNA. It is just cutting hydrolysis of a phosphodiester bond and creating a phosphodiester bond. This is what recombinant DNA technology is all about. If one understands this, I think everything else is a child's play as far as recombinant DNA technology is concerned. Here are several such enzymes. I mentioned about EcoR1. This is from another bacterium called Bacillus something, amylophysians or something like that. This is Haemophilus aegyptus. This is Haemophilus hemolyticus. This is Xanthomonas something. You see all these sequences that you see, they're all palindromic sequences. In the word Malayalam, either you read it from forward or the backward. So there's just a symmetry, a diode symmetry of axis that these enzymes actually recognize and will cut. Either they will cut between this or they will do right in the middle. So depending on the enzyme, you have ends, for example, you have something called the sticky ends. You can see these are sticky ends. The ends are not, whereas the other enzymes like this, they cut right in the middle. So it is a blunt end. So enzymes do only one of the two things. Either they create a sticky end or a blunt end. But the ligase will join all of them if you provide the necessary cofactors. Now, once you take a DNA, if you take a fragment of DNA or a plasmid, as we call an extra chromosomal element, or any DNA, for example, and use a battery of enzymes, or use an enzyme called A, it will chop it off into fragments, which can be separated on agarose gel. By an electrophoresis method, you can separate the fragments. Then you can cut out this fragment, extract that fragment, and then do all manipulations that you want. This is another enzyme on the same DNA, but cuts in a different way because the sequences are different. Then a battery of another enzyme called C. So by gel electrophoresis, a pattern of restriction digestion can be created. And if you use all the enzymes that you have, many enzymes, you can actually create a whole map. This is called a restriction map. By using a variety of restriction enzymes, you can actually make a restriction map. And this is the first thing that people do in genome sequencing. When you want to sequence, for example, the human genome, which was sequenced in 2000, was actually cut up. It has 3 billion bases, and it was cut up with a lot of restriction enzyme, lot of pieces together, and then you sequence each of these pieces and then join together, and you have the whole sequence. So you go back now to Daisy Duswas and Werner Arber's experiment. The discovery that these enzymes exist in bacteria, and you see the applications of that discovery, is phenomenal, and certainly a Nobel Prize winning work that Werner Arber and the other two, Dan Nathans and Hamilton Smith, were actually awarded in 1978. Now, I'll show you one more slide on research. This is an organism that my laboratory works, Helicobacter pylori, causes cancer, is the only bacterium that causes cancer in humans. It causes ulcers and it causes gastritis. And I couldn't put all the things together, but when you sequence it, you have a lot of information. And each of the, by bioinformatic analysis, you find that these open reading frames, which actually probably code for a protein, there are 44 or 45 of them. Now, if you go back and just think about it, Werner Arber's discovery, restriction modification is basically a defense system. When the phage comes in, the phage is not welcome, and therefore it has to be chopped off. And that is the main purpose of RM systems as far as the bacterium is concerned. Why have 44 of them in Helicobacter pylori? And Helicobacter pylori is an organism that survives in acidic conditions, found in the stomach, where the pH is almost 1 or 2. How do you imagine phages occurring at that pH? And therefore, the interest in my lab was to find out what do these RM systems do. But that's a different story that is not a Nobel Prize winning work, but some very nice findings that were found as to why these systems actually exist in large numbers. So coming back to phage and bacterium. So the phage, which is a bacterial virus, has first to adsorb onto the bacterial surface. So there are receptors for that. So you can do it goes through adsorption. I'll just go through this cycle here. Sorry. So it's adsorption, and then the DNA is injected, and that phage DNA is replicated, is made into 2 copies using the host machinery. Then the phage DNA is replicated, that is transcribed and translated into RNA and proteins. And then the smaller, baby phage molecules are made. A lot of them are made, and then it kills the cell so that the phages come out and they can go and infect another thing. So phages are very, very powerful. A phage infection is something that many biology labs can actually do, which do also that, and to show students that this is the typical cycle of a phage replication cycle. So you can actually prevent phages at the adsorption step by blocking the phage receptor, by producing extracellular matrix proteins, which will probably inhibit this entry. You have something called the abortive infection system. And in terms of preventing the phage DNA entry, you have the restriction system that I just mentioned about. This was done in 1960s. And now in 2007, 2005, 2007, again we come back to phages and DNA. And another system that all these bacteria have is something called the CRISPR, which is the topic of this year's Nobel Prize in Chemistry. That the discovery of the CRISPR-Cas system, not just the discovery, but the use of CRISPR-Cas system for genome editing, is the subject of the Nobel Prize in Chemistry this year. What I want to emphasize here is we are back to very primitive systems, the prokaryotic systems, that is, the bacteria and phages. And it's true now, we have learned over the years that is what is true for E. coli or for a bacterium is also true for the elephant. All the biological processes that people study all the basis for that, everything has come from the study of bacteria. And I think, this has to be emphasized, especially in our country, that research in prokaryotic biology becomes very, very important. So this is a brief introduction to the CRISPR system, and I'll tell you what is the CRISPR system and how it is used for genome editing. But the whole idea of CRISPR is actually a part of the biology of the bacterium and the phage. While the RM system is an innate immune system, the CRISPR is an adaptive system. And that is true even in us human beings. For example, our human immune system can be easily studied under innate immune system or the adaptive immune system. But this is true even for the bacteria. It was always true for the bacterium that we have here. Maybe I should just find out if everything is is going on well. Is there any interruption, anything that is not clear?
No, everything is very smooth so far.
All right. So I will continue and then again ask you after 10 or 15 minutes if there's any
Sure,
problem.
sure.
Now, this is a very simple slide. What is gene or genome editing? What is it? What are we doing? You remember the background, huh? The bacteria and the phage, the immune system, the innate immune system, and the adaptive immune system.
Right.
We'll come to something else, and I will connect the two later on. So the genome editing, which I've written here, as the Germans call it, the Gedanken experiment, the thought experiment. The objective of the experiment is targeted replacement of a mutant gene. Here you have, this is the red one which is shown here. And you want to replace this mutant one. There's some mutation in this gene, and you want to replace it with the wild type or the normal gene or the healthy gene. And this is what is genome editing all about in a very simple way, absolutely simple, simplistic terms. You're converting that red arrow to a green arrow. This is a mutant, this is a normal gene. When you do these kind of experiments, how you do is what we are going to talk about by using this CRISPR system of a bacteriophage and bacteria, that you can do this very nice experiment. And if you do it in germline cells, that is, in the embryo or in the sperms and things like that, you can produce transgenic organism because from one generation to another generation, it can go on. Let me tell you right at the beginning, these experiments in germline are not allowed. The NIH regulation system does not allow anybody to do germline experiments, except there was one Chinese case, which I'll talk about. He was condemned later on for doing this experiment. Most of these experiments, gene therapy is something that you have heard. This is a simple gene therapy experiment. Is done only in somatic cells, like skin fibroblasts and things like that. So essentially, the objective of this experiment is targeted replacement of a mutant gene with a normal gene in the human cell. And in a converse experiment, one could replace a normal gene with a mutant allele. Allele is a gene in one cell, essentially knocking out that gene in the cells. Why would you do a reverse experiment? Just to find out what is the function of this gene. There, although the whole genome is sequenced now, the human genome is sequenced, there are many regions in the DNA we don't know what is the function of this. And therefore, if you knock out that particular gene and then see what is the effect of knocking out this gene, then you can actually ascribe a function to it. Not only in humans, but in plants, in bacteria, and things like that. A number of these open reading frames I told you about, we still don't know what they code for. And therefore, knocking out or removing them or inactivating them, that will result in a certain something called a phenotype, and from then we can ascribe a particular function to it. So this is the Gedankt experiment on genome editing. So genome editing is a process of introducing specific modifications in the endogenous genomic DNA, including you can delete a region, you can do point mutation. A G can be replaced by a C or whatever it is. You can introduce inversions, you can use translocations. These are varieties of manipulations of DNA. And by using something which I don't have time to talk about, homology directed repair, it can also be used for genetic correction. Now, gene therapy is all part of this. And why do you do genome editing? To understand the function of a gene, a region of DNA which transcribes to RNA, which then translates to a protein in a sequence specific way and monitor its effect on the organism. And especially when you study bacteria, you're studying growth, for example. I knock off this gene and then try to see the growth kinetics of it, and therefore distinguish it between the normal and the mutant gene. For example, there are 20,000 or 25 or 26,000 protein coding genes in the human genome, and we still have very little idea of what at least 6,000 of them actually do. Now, actually, I could knock off one of them and then see what happens to the human being, but such experiments are not allowed. And therefore, we go to mouse systems or yeast systems where we can do some of these things. And many of these actually correlate with human development and things like that. So that is the purpose of, and this is genome editing and why we do genome editing. Now, you remember the prize, the foundation gave the prize for a method of genome editing. So there are a number of methods, but the most common, the three common targeted genome editing systems are the zinc finger nucleases. I'll just go by these names, please, and I'll explain to you later. Something called TALENs, which is something, I'll tell you what it is. It's a transcription activator like effector DNA cleavage domain and things like that. And then you have something called the RNA guided nucleases, and that is where the Nobel Prize has been given for use of this to do genome editing. But I think it's very important for us to realize that the previous two, the zinc finger nucleases and the TALENs, actually were the first genome editing methods. And many of us in this area of nucleases, like me, who study, know the kind of sensation that was created when zinc finger nucleases were created and how this had the potential to do genome editing. And I spent a few minutes on the zinc finger nucleases simply because of a personal reason, and I'll tell you what it is later on. Now we come back to restriction enzymes, that enzymes that recognizes the DNA like EcoR1 and cut the DNA at the G and A, and you get two pieces of fragment. Many enzymes do this. Many restriction enzymes do it. But there are some enzymes called restriction enzyme called the type 2S. And these enzymes also recognize a specific sequence, right? But they cut slightly away from they don't cut within the sequence, but they cut away from the sequence. About 9 nucleotides or 14 nucleotides away from the recognition sequence. So these are known as the shifted cleavage enzyme. The type 2S, the S stands for cleavage, the shifted cleavage enzymes. And obviously, when you have these enzymes, that means action at a distance, right? They act here, but they bind here, but they act at a distant region. Like you cut here, most enzymes cut within the sequence, but these enzymes recognize the sequence. They're very specific, but cut away from the sequence. And therefore, there's very nice work. This was done in the 90s by an Indian origin scientist, Srinivasan Chandrasekharan at the Johns Hopkins University, from this bacterium called the Flavobacterium, something it's called, Ochinidis or something. He studied this enzyme in detail. He was a chemist from Pondicherry, went to do his PhD at the Georgetown University in Washington, D.C., and then moved to Johns Hopkins to work on this FOC1 endonuclease. And he and his group figured out that the protein has actually two domains. One part of the protein recognizes, it's called the recognition domain, and one is called as a nuclease domain, which actually is responsible for cutting the phosphodiester bond. And these two domains were linked by a linker region here. And by using very standard biochemical methods, he separated these domains and showed that this nuclease domain by itself can cut DNA, but in a non-specific manner. But the minute you fuse it with the recognition domain, it would cut very specifically. So he actually split this protein into two portions, one called as the DNA binding domain, that is where the DNA binds, and one is the nucleus domain. In 1993, at a FASEP meeting that I attended, this work was actually presented. And what Chandra had done at that time was to take different kinds of DNA binding modules, nothing to do with the restriction enzyme. There were a lot of other modules called the zinc finger modules, which are all DNA binding. And as you see here from this picture, this is one module which recognizes this sequence. Here's another module of zinc finger which has this. The zinc finger is because the zinc is essential for activity. It actually helps the protein to fold in this way. So by using various modules of the zinc finger types, and he fused it with the nucleus of the FOC1, and lo and behold, you created an enzyme that does not exist at all. You're actually playing God here, and you have created a new enzyme altogether. The beauty of Chandra's experiment was that he could now create a restriction enzyme which would cleave at sequences that you wanted it to cleave, because you have a variety of sequences that the zinc finger modules actually recognize. So by fusing the various modules, you can create a nice restriction enzyme and actually cut it at the place that you want. And that is what genome editing is all about. And this was a paper that he published where the first time the word gene editing or genome editing is actually used. For many in this area of this, everyone, like including me, feel that Chandra should have got or should have been recognized more than what he has been recognized. But the Nobel Prize citation is very clear for a method of genome editing. But the zinc finger nucleases were the first gene editing nucleases, and this whole concept is because of Chandrasekharan's work. I thought I must mention here. He's also a good friend of mine. He spent 2 years, about a year in my lab, and we ran some genome editing workshops and things like that. I'll skip this slide. Soon after, this was in 1997, 99 or something like that, and in 2009 now, these another set of proteins from another bacterium called the Xanthomonas bacterium that infects plants, the people isolated these DNA binding domains called the TAIL, called the transcription activator like effector proteins. You must bear with us, the biologists, because we love to give acronyms for these kind of things, and therefore we have plenty of them. And the nuclease, if you now take this tail, the DNA binding, just like the zinc finger, and fuse it with the FOC1 nucleus, you have a nucleus called the talons, which is shown in the next slide. Right? So this is what talons is about. You require two talons, talon on the left side, talon on the right side. The FOC1 nucleus domain is there, and it would cut DNA exactly where you want. This is a second generation of genome editing nucleases. I think the zinc finger nucleases were the pioneer nucleases, and once the talons came, just about the time, actually, if you go back and read the literature, this is in 2005, 2004 or something like that, this whole phenomena of adaptive immunity, which I talked about, was actually studied. Actually, a summer student in Spain was analyzing the sequences, the genome sequences of a bacterium, one of those halophiles, which especially on the coast of Spain. And he found in the genome of this organism a lot of repeat sequences. A repeat sequence with the same sequence repeated, but there was a spacer between the repeats. Something like this, as is shown here, that you have that one, two, three. So these are the repeats. The unshaded, uncolored ones are the repeats. And between the repeats, you had something called a spacer, which was completely a different kind of sequence. Whereas the repeats were identical. The repeat before one or before two or before three were identical sequences, nucleotide sequences, and about 21 to 48 base pairs. The spacer length was 26 to 42 or 72, and these were entirely different. So this pattern in the genome fascinated this young summer student, and he was working in a laboratory in Spain, the University of Alicante, I think, where the existence of these repeats and spaces, and the term CRISPR was actually given by these people, called the clustered, regularly interspaced short palindromic repeats. That is what CRISPR stands for. Subsequently, a few years later, this CRISPR locus, or locus is a region in the genome, always had these genes called the Cas genes, called the CRISPR associated genes. There were a number of them: Cas1, Cas2, Cas3, Cas9. And the famous one is the Cas9 that we're going to talk about. And this had a nucleus function. That was, it was able to hydrolyze a phosphodiester bond. So the existence of the CRISPR, the sequences in the bacterium, followed by a nucleus and enzymatic function, gave rise to this whole adaptive immune system of the bacterium that people studied. And that is what Emanuela Charpentier's work is, to describe this CRISPR-Cas system. And I'll come to it a little later. So the CRISPR-Cas system is found in 48% of bacteria, 90% in archaea, which is between the eubacteria and eukaryotes. And this is the general arrangement. Okay, so you must remember this. You have a repeat, you have a spacer, you have a repeat, you have a spacer, you have a repeat. And the whole genome is full of this repeat-like sequences here. This is the fundamental observation that was made by a summer student way back. Actually, even before this guy's observation, Ishino in Japan actually made the observation in 1987 in E. coli. But then he dropped it, and then it was picked up later on by these guys. So I will describe to you the CRISPR-Cas system, which is an immune system, an adaptive immune system, which naturally occurs in a bacterium. Many bacteria have this defense system. The restriction modification system of Werner Arbor is one defense system. This is another system. So let's go this. I'm going to go through this very slowly so that you will follow it. So you have a page here which absorbs and injects his DNA, right? This is the DNA of the phage here. This is recognized by those Cas genes of the bacterium called the Cas2 and the Cas3. They bind it and bring it all the way to this CRISPR locus, where you have this repeat, spacer, repeat. And in the spacer region is where this DNA is actually incorporated, right? Now, so you now have a new CRISPR details. You have a new spacer now that is formed here. This formed here. So you have the Cas genes here, and you have the CRISPR locus. This is the DNA element that we're talking about. These are the proteins which actually are helpful in the whole genome editing that we talked about. Emanuela Charpentier's work, she was in Europe all the time, but she had gone to Yale for some postdoc and came back. But in the University of Umeå in Sweden is where she discovered this element here called the tracer RNA, the TRACRARN. And I'll come back to this later on. So this now, when the phage infects, and if it's not chopped off by the host DNA, our host system, this is what happened. A phage, a small element of the phage genome is incorporated into the bacteria. It's integrated into the bacterial genome in between the two repeats that we talked about. The black ones are the DNA repeats. Now, if the phage now infects again a second time, again it injects a DNA. But this DNA is now recognized by the bacterium as something which is not to be allowed. And this is the adaptive immune system that I'm talking about, that this region of the bacterium now, this one here, which is shown here, is actually transcribed, where a DNA molecule is made into an RNA molecule. The RNA molecule is now consists of spacer, repeat spacer, repeat spacer, repeat spacer. It's called the CRARN. This is chopped off into small pieces. This combines with the enzyme called the Cas9, which is there. The Cas1 and Cas2 are the first ones to recognize, but the Cas9 is the nucleus that we're talking about. Cas9 combines with the small RNA molecule that Emmanuel Charpentier discovered. This RNA molecule combines with Cas9, and then it recognizes this particular one because the sequences are already there, and therefore there will be homology, and therefore they will bind. And this complex is actually the one which recognizes in the phage now, and I'll show you in the next couple of slides how this recognition occurs, and then the scissors actually chops off the foreign DNA into pieces, and therefore the bacteria are resistant to the virus. This is the adaptive immune system. The innate immune system that I talked about is methylation, preventing restriction to occur, but then this is the adaptive system, and therefore the cleavage of foreign DNA is possible. So three things are important as far as the CRISPR-Cas system, which is the naturally occurring bacterial system that you have here. This is the same slide. I have 4 or 5 different versions. This is from the Royal Swedish Academy of Sciences, which actually gives the chemistry prize. You should go back and read this, go to the site. This is beautifully explained here. The same thing that the phage DNA injects here, the viral DNA. The viral DNA is actually, if it's already there, it is recognized by this. The CRISPR DNA is now made into an RNA molecule, which combines with this tracer RNA, which is part of the system that you have, that Emmanuel Charpentier recognized, combines with the enzyme called the Cas9, and this together combines and to form a genetic scissors, now acts on the foreign DNA and then chops it off into pieces. In other words, this recognition and enzymatic action is the most important thing. And this is where Jennifer Doudna's contribution actually comes in. She worked out on the mechanism of how the CRISPR-Cas system actually works. And I'll tell you briefly, although I still have some time to show you that. So in her studies, so you have the tracer RNA, which is the Emmanuel Charpentier's molecule. After this, they met actually in Puerto Rico, and then they decided they will combine and then formulated this whole system. And this is the science paper in 2012, which is the paper that is cited for the Nobel Prize. So you have the Cas genes here. The next few slides are all repetitions, but I just want to drive home the point that these are the molecules, these are the players of this CRISPR-Cas, the TRACR-RNA, the Cas9, these are the Cas genes which recognize, and the CRISPR RNA, the repeat spacer, repeat spacer. This particular piece of DNA is transcribed to make a pre-RNA molecule, which then is chopped off, which combines with Cas9, which is already combined with the tRNA, and then this acts as an RNase molecule. So this is an RNA-guided nucleus, not just DNA, but it's an RNA-guided nucleus. And this the other molecule that I was talking about, actually helps to recognize a sequence just upstream of this called the PAM sequence, and this is specific for each enzyme. The Cas9 is one version of an enzyme. There are other nucleases that people have used, and I'll come to that later. And that recognition is done by this. So you have this whole very nice structure that is formed here. The genomic DNA, the RNA, which is transcribed, and then this kind of a molecule, which then where the Cas9 actually works, and you get cleavage. I'll skip this. So this is what it is. I've written as introduction to CRISPR, but this is what it is. The CRISPR locus, you have the short palindromic sequences here. The pre-RNA molecule is made. It's actually chopped up. When the RNase 3 comes in, you have to chop it up into small pieces here. This combines with the Cas9 and the other RNA molecule, which is the complex that is formed here. And that then recognizes the Cas9, then chops it up into various pieces. And detailed mechanism is actually worked out by a number of labs, but I think Jennifer Doudna's lab contributed immensely for understanding this. More than anything else, she used the system to do actual genome editing work, which I'll talk to you about a few slides from now. So the Cas9 is a nuclease, is an enzyme that people have. All the crystal structures, all the crystallography has been done, and therefore, because of crystal structure, with and without the RNA molecules, we actually know how this Cas9 is actually able to cleave the edge. So this is the whole complex thing that I talked about. So it's an RNA-guided DNA cleavage by this enzyme called Cas9. This is the most commonly used now in genome editing. This is from an organism called Streptococcus pyogenes. A number of bacteria have different versions of Cas9, but this is the most useful one or easy one to use. The Cas9, people are trying to find out smaller molecules and smaller than Cas9 from other bacterium, and that is the kind of work that many people are now engaged in to do these kind of things. So this is what is important here, that this nucleotide target, the protospacer, and the NGG, which is called the PAM sequence, which is shown here, are the important regions for recognition. And therefore, if you now synthesize RNA molecules to the region where you actually want a cut to be made, you can get precise cutting at that point. It's not random, it's not haphazard, but exactly at the place that you want to cut DNA, you can monitor this, you can engineer this. There are two RNA molecules, and therefore now people have actually combined the two molecules and known as the guide RNA molecules. And by using a suitable guide RNA molecule and the Cas9, you can go to any region of the genome and make an actual cleavage. So this is what is the CRISPR-Cas, and the exploitation of the immune system to actually do genome editing is what Jennifer Doudna has done very successfully. And a number of other labs also have done this, and I'll come to that. But I think the actual exploitation of this to do genome editing is what is done here. So what you do in the CRISPR, how do you, so this is a system. So you have a CR-RNA and a tracer RNA. Now people have actually, you can design and make one molecule, and this one as a chimeric guide RNA molecule. You have the protein Cas9, which is now available commercially. You design the cRNA to target any sequence next to the PAM sequence, which is the recognition sequence. And then the Cas9 creates a double-strand break in the genome, exactly at the place that you want to cut. And the double-strand break occurs on both strands, and then this is actually repaired by a recombination process, which naturally occurs in all cells. And then you can get the place where you want a change to occur. This is exactly you can do. So this is genome editing very precisely by exploiting this particular system. I will not go to this, but just to compare the three gene editing nucleases, the ZFNs, which were done by Chandra and his group in Johns Hopkins in 1996. The Talons came 2010 The CRISPR-Cas9, the science paper that Jennifer Doudna's and Emmanuel Charpenter's is 2012. Here's all the details of comparison. I think the most important thing to remember is the issue of specificity. That the CRISPR-Cas system is very, very specific, more specific than the zinc finger nucleases and things like that. In spite of understanding the whole mechanism, there are situations where either the CRISPR-Cas or the zinc fingers or the talents can cut away from the target. And we still don't understand this off-target cleavage. And therefore, one has to worry about this before you can use these to apply in human therapy or to food sources, because you don't want other regions of the genome to be chopped off. And therefore, these experiments are done as far as humans are concerned in cells, and then you put back the corrected cells into the human being, and ex vivo kind of experiments. But you can't do germline, you can't do it in the eggs or the sperm DNA or the zygote DNA. But if you compare these three techniques, as this is summarized here, the target efficiency is very, very high in CRISPR-Cas, but not perfect, not 100%. Whereas this is low in zinc finger nucleases. So these are the three methods you have now. There have been a lot of success with zinc finger nucleases with talents, but I think with the CRISPR-Cas, it has absolutely overshot everything else because it is so easy to use, very, very cheap as compared to zinc finger, because it's all protein-based, thing. Whereas here it's an RNA, and RNA is cheaper than a protein in many sense. And therefore, I think the popularity of the CRISPR-Cas, because of its simple way of doing things, has really popularized. So you go back in 1987. This is just to summarize the history of CRISPR, just the CRISPR. We're not talking about zinc finger nucleases. The first report was done by the Japanese scientists who saw these clustered repeats. And as you go through 1987, through all these years, this is where the Jennifer Doudna paper, Emmanuel science paper came out in 2001. So from 2012 to 2020, it's almost 7 or 8 years. And it's a very, very quick recognition. Although they have got a number of awards for this work, the Nobel Prize in Chemistry is a peak in their careers and things like that. And if you go back, this whole story of the CRISPR, it actually starts off in Spain, where this was done in a place called Alicante, where the boy actually saw these repeat sequences. And then all over, you have people now taking up in such a way that, it's become, there's a craze for the CRISPR thing, because the Cas9 can be programmed to perform gene editing in mammalian cells. It is simpler and easier than other genome editing technologies, and an unprecedented efficiency and stunning ease of use. Even when Chandrayaan came, we did a CRISPR experiment for a workshop here and showed that this is easily done. If we look at the market, for example, there are about 150 or 170 companies which make CRISPR-Cas, or they do this CRISPR-Cas genome editing. And you can see here from 2017, and it's expected that in 2025, you'll have a huge market in the CRISPR-Cas9 technology. Number of publications, as you see over the years, it is just shooting up, because every kind of, in agriculture, in livestock, animal husbandry, medicine, the CRISPR technology is used, because you can edit the genome and make better species of rice and things like that. In agriculture, it is heavily used, and you will find this publication. As far as patents are concerned, I think it's important, because till recently, there was a huge battle on this patent landscape between Jennifer Doudna's lab at University of California and Berkeley, and other scientists whose picture I'll show you later is Feng Zhang at MIT Broad Institute. And the patent was finally given to the MIT and University of California at Berkeley. Jennifer actually lost the patent. Right, there's some application of that, and you see the number of patents that have gone up in the last few years here. You see that. So this has been a wonderful story, but everybody should remember that this is a method of genome editing. There were the zinc finger nucleases first to be described. All three of them do the same thing. Either you edit the gene, you add the gene, remove the gene, you disrupt the gene. All these do it, but the efficiency varies from one system to another system. Here are some examples. This is done by either zinc finger nucleases or the CRISPR. I forget now. You can make mushrooms, for example. You can make white button mushrooms, which will resist to browning. Most mushrooms, if you keep oxidized, and you can prevent that. You can introduce color to lovely flowers, beautiful flowers you can create by actually manipulating or do genome editing. And one, you can make cows, for example, without horns, for example. And this is a big market for hornless cows and things like that. Here is a very nice experiment that came last year, 2019, where they edited a gene called LSDR1 in natural dextral clockwise pond snails, which was right anti-clockwise and clockwise. You can actually reverse it by changing one base in this LSDIA gene. So lots of things are possible here. Handedness, left-handedness, right-handedness, so many interesting problems that you can study by using this CRISPR-Cas technology. Here's the best example. This is also not done in human germline cells, but in somatic cells, for example. You have a patient with a disorder, sickle cell anemia or, thalassemia, so many diseases, You can take the patient with a disorder, you isolate these cells, which are immune cells, and the other cells where you want to do this. By genome editing, using gene editing nucleases, Chandra has actually done with the zinc finger nucleus, but it's much easier to do with the CRISPR. You can change that and then put it back into the human being who is suffering, and then you rectify. So, in one sense, gene therapy and these kinds of ex vivo, not in vivo, but ex vivo genome editing approach for blood disorders is where there's a huge market, and not market, there's a huge demand, and CRISPR-Cas technology has really gone a big way in this. Most importantly, actually, and I want to bring this point here, it's actually the yogurt industry, the yogurt manufacturers. In the early 2000s, they did a lot of research on this. This bacterium called Lactobacillus acidophilus is very important for yogurt industry. And yogurt industries had a lot of problem because bacteriophages were coming and ruining all the cultures, the mother cultures, everything was ruined, and there was a huge loss. So this company called Danisco, which is now acquired by DuPont, actually did a lot of research on these bacteriophages and bacteria, and came out suggested that that some of these things can be addressed by CRISPR immune system to protect important bacteria like this, which is used in diet and yogurt industries. More recently, Feng Zhang in MIT Broad Institute in Boston actually has developed a nucleic acid detection system using the CRISPR nucleases. It's called the Sherlock. It's a rapid detection of nucleic acids, and it's integral to applications in clinical diagnostics and biotechnology. And they use this CRISPR technology to show this. And now we in India have also a CSR institute called the IGIB, and with the Tata Group have developed or used the CRISPR system to call Feluda. Feluda is actually named after Satyajit Ray's famed detective. Again, the same like the Sherlock test, who uses the CRISPR-Cas technology to detect genes specific for the the COVID virus that we're talking about in the pandemic region. This is a much cheaper one. I'm told it's priced at 4,500 rupees. It takes 45 minutes, but now people all want less than 15 minutes, less than 5 minutes. And therefore, CRISPR technology again has come in a big way uh, to this. Now, the last, Jayanti, do I have a few minutes?
Yes, sir. Please go ahead. Yeah.
All right. This is all I wanted to tell you about the CRISPR technology and the work of Jennifer Doudna on the left and Emmanuel Charpentier. This is when they received one of those many prestigious awards for the development of a method of genome editing. But I think the next slide I want to be is that who gets the credit for CRISPR? While most prestigious awards in the country, in the world, have given out to Emmanuel Charpentier and Jennifer Doudna, I want to bring two people's work. This is a friend of mine, actually, Virgis Siksenis in Lithuania, who actually published or had the work done, but the journal Cell delayed his publication 6 months, which enabled Emmanuel Charpentier and Jennifer to publish a science paper. 6 months later, Virgis Siksenis' work came out in the proceedings, the PNAs of the United States Journal of Science Journal. Virgis Siksenis actually shared the prize, a more a more rewarding prize called the Kavli Prize, with the other two, with Emmanuel Charpentier and Jennifer Doudna. So this is one person I think, it's a real letdown. Everybody thought that Virgis will share. The Nobel Prize is usually can be given to 3 people, but this year they decided to give it only to 2 people. But Virgis Siksenis' work is absolutely original. It was 6 months later, 3 or 4 months later, it was published. It was delayed by Cell. They didn't even review the paper. They just rejected the paper. And Virgis had to go to a number of journals before it went. And in the meantime, this was actually fast forwarded. The work of Emmanuel Charpentier, it's a fantastic work, but when you submit the paper, and that is very important and things like that. The other person here, which I've shown here, is Feng Zhang at MIT Broad Institute in Boston, the one who now shows this Sherlock test. His contributions in the CRISPR-Cas mechanism is also outstanding. Not the mechanism, but the use of it in genome editing. And another colleague of his, George Church, is another name that you normally associate with the CRISPR-Cas system. So I think, Virgis Siksenis, George Church, and Feng Zhang are the other thing. If you now read science magazines like Science and Nature, you'll find a lot of articles on how these guys. But Feng Zhang actually got a patent for the CRISPR-Cas technology. Especially this year, after the prize was awarded, there are a lot of reports that I've read. And one is the soaring popularity of the gene editing has made celebrities of the principal investigators who pioneered the field. But the graduate students and postdocs are often overlooked. And I think in all the 3 groups of Jennifer Doudna, Emmanuel Charpentier, Feng Zhang, and Virgis Siksenis, the first author papers of this are again doing so well. They've all become independent, and there's a lot of talk this year, especially on this. The first authors of these papers should also be recognized in some way or the other. I thought these are called the unsung heroes of the CRISPR-Cas system. Jennifer Doudna actually credits a laboratory success to someone she took a chance 20 years ago, a laboratory assistant called Kai-Hong Zhu. And I think they get along so well that every success of Jennifer Doudna, she says, is because of this Kai-Hong Zhu, who was the lab manager. That reminds me of another thing of a person called Daisy Duswa, who was with Werner Arber, who was a laboratory assistant who actually did the experiments, was the first author on the paper, And there was a lot of talk that when the Nobel Prize was given to those three for restriction enzyme, Desirazu actually missed the prize. Should have been cited. But her bad luck, after she left Geneva, she went to University of California, San Francisco, where she worked with Harold Warmers, who also got the Nobel Prize. But again, Daisy Deuce was work or contributions were not there. So she moved back to Paris, where she served as a group head of a mycoplasma laboratory at the Pasteur Institute. And in 2014, she passed away. So I think, there are people, every prize, I think there are others who also certainly deserve it. It's not that the people who got didn't deserve it. I think they certainly deserve the prize. And I want to conclude this talk by a couple of slides here. The Nobel Prize in Chemistry this year honors the work for the development of a method for genome editing. It's actually, a crack in creation, gene editing, and unthinkable power to control evolution. This is the title of the book that Jennifer Doudna actually wrote a few years ago. I'm trying to get this book. I think it'll be a nice book to possess. It's a very nice thing. And I think, last year, 2 years back, and all the recent chemistry prizes, it may seem that the chemistry Nobel has again been biologized. Mostly biologists are getting, biochemists are getting. It's sometimes hard to see how an enzyme or a phage are chemistry, but they are. It is the chemistry that governs the shape of these molecules, which in turn affects their function. And I think, this is what Frances Arnold said 2 years back when she got the prize, that chemists are inspired and awed by the biological world, but what they should really be inspired by is the process by which biology discovers chemistry. And that is called evolution, which is typically biology. So I think physical sciences are actually the drivers of biology in health. And this year's chemistry in Laureate's work shows how studies on basic principles in biology, the adaptive immune system, can have profound implications for technology and medicine. And it again demonstrates the critical importance of continued funding of basic biomedical research and not all the time harp on applied research, applied research, applied research. But more than that, I think it is the interdisciplinary nature or approach to science that is very important. And as many people said, this is quoted from somebody, invest in long-term blue sky science, in people, in ideas and teams to take the risk on infrastructure and let people follow their ideas and their dreams. And this is exactly all the Nobel Prize winners so far have actually done. I finally want to conclude that there is an arms race between bacteria and bacteriophages. I won't read this, but I'll show you the last slide that when life started, bacteria were very happy. They're all smiling here because the phages could not enter the bacteria because of restricted systems, and the phages were unhappy. But as evolution progressed, as life progressed over the years, millions of years, you now have bacteria which are not smiling, whereas the phages are smiling because they have this anti-restriction. And just to tell the audience here, they have now a lot of work on anti-CRISPR systems that people have actually found out. So I think I'd like to emphasize study very simple systems like bacteria, and you have a wealth of information that is created. And what is true for bacteria is also true for the elephant. So I'll stop here and take any questions. Thank you.
Thank you so much, Professor Rao. It was a very, very interesting talk. I can understand with the real complexities involved how much
Yeah.
you had to really process it for the audience. Thank you so much.
All right. You're welcome.
You can start your video. Yeah, it is on, I think.
Yeah.
Okay, but the whole screen is so maybe you have to unshare your other screen.
I have to do this.
And,
Now,
and
how
then,
it's always a problem of putting on, and everything has gone blank now.
yeah, okay.
How do I put it on? Nothing is
Come in.
coming.
Any indications?
So I say unshare, no?
Uh, yeah, I'm not that familiar. Uh, okay, I think some things have been disabled. Naveen, is there a processing that you have to do?
Stop share. Varna, Varna.
Is
Perhaps
my video on?
So you could say stop share, sir.
Okay.
All right.
So you can say stop share. So
it has stopped.
You-
Sharing has stopped.
It just stops.
yeah.
Okay. All right.
Okay, but your voice is very clear. Shall we start with the questions?
Yes, please.
Okay. Uh, this question is from Harshit. The question is there any limit of genome editing? Like which mutant gene can be put on which gene? Like can we put an elephant gene in a mouse? And how will that
No.
change the host?
Yeah. No, no. I think some things it's all nice to think about. Maybe in the future, but right now, many things are biology will not allow that. But the systems all have a very inherent way of checking things, so it's
Okay.
not that simple. But genes which look similar, yes, you can certainly you can put, but it'll be function as the same gene.
Okay.
Eukaryotic genes or higher animal genes and all, they have all lot of modifications and things like that called
Okay.
post-translation modification, and therefore those things affect the function. It's not just the DNA, but the protein that is made, the protein is modified and then it functions as it's supposed to function. Yeah, but technically and academically, yes. But
Not at there. Yeah. There is a question from Manomi. The question is there any specific reason why the experiment is only performed in the somatic cells?
is what specific for somatic?
Why the experiment is only performed on somatic cells?
Because germline cells, it will go on to the next generation and all the future generations. That is not allowed by NIH.
Okay.
Even gene therapy, what they did before they stopped, I think they've started again. It's only on terminally ill patients, but
Okay.
not on regular patients because of ethical condition and ethical considerations.
Well, will there be a modification of those rules once we understand things better and we are able
Yeah,
to?
yes, we should. Yeah, I think so. Yeah.
Okay, Madhav Nakani has actually asked that question about Feluda, which you have already
Good.
discussed. Okay. Uh, okay, so there is another from Madhav, which says uh, the ethics part of the tool. How does Professor Rao see it evolving over time?
Yeah, I think there should be a lot of discussion, especially the ethical and regulatory things. Regulatory bodies are very, very finicky about this. You can't, there was, I mentioned in my talk that a Chinese
Chinese,
scientist actually did this, he edited three babies were born after editing. And I think there was an uproar. Even in China itself, he was actually condemned. So it's possible to do these things, but you're not you need a lot of clearance, you need ethical clearances which he never got. I forgot his name starting with J, but I think he was completely condemned for this. But if something happens like this, that is why the technology is so simple, now that Charpentier and Jennifer Doudna actually made it so simple that you can do it.
okay.
um, there are cases where you have actually de-extincted animal of species. You know that
Oh,
an animal of species which was extinct, they brought it back, but luck was fortunately, unfortunately, it died. So it got extincted a second time. So you have cases like this.
yeah.
These are all Jurassic Park stories that are all possible. But a long way off. So regulatory bodies are very, very important too.
Okay, there is a question from Arun. Uh,
Ah,
Sir, the way the bacteriophage attacks the bacterium cell is very
ah.
similar to how the HIV virus inserts its genes into the human genome. Can we not devise a similar RM system to develop a cure for AIDS?
Yeah, yeah. I know RM systems. People have tried, but they have all failed for a variety of reasons. When you inject, or if we try to put these, I think the half life, the degradation of these proteins is very fast, and I think people have tried some of these things, of especially AIDS, by using another set of enzymes called the ribozymes, the
Okay.
RNA enzymes, but they were never successful. For a variety of reasons, which I don't remember now, but I think people tried.
Okay. So there's a question from Richard. Why CRISPR is found only in 48% of U bacteria? Why in the other species, or why not in?
In archaebacteria, it's a lot of archaebacteria. Mostly it's archaea. 85% of archaea have the CRISPR system. The
Okay.
U bacteria don't have. No, there is, but it's 40 or 30% or something like that. I don't know why it is there. Is what is found.
Okay. So, yeah, are there any more questions, Naveen and Jayandi, Rashmi? I have actually a question. You
Yes,
mentioned a sort of legal battle
yeah,
for the patent between MIT and Berkeley recently, which
right.
MIT won. I suppose it was a very big battle. Is there a lot of money or a lot of big stake involved in that particular patent?
Yes, yes, billions probably. Yes, yeah, millions of dollars. Yes, that
Okay.
is right. Yeah.
Yeah. How good is our own patent regime with regard to these biological processes?
Which one?
In India,
Ah,
patenting system for biological processes.
no, it's picking up. I think I find even the institute for like mine, the institute in Bangalore. The last 10 years, I think we've been worrying about patents and things like that. But
Okay.
before that, we never bothered so much. But now the awareness is there. The
Yeah.
Delhi, the IGIB, which is doing this Feluda test, has actually probably done some of these things.
Okay.
But again, Feluda is based on Sherlock, which is Feng Zhang's work. So the
Okay.
idea is not particularly origin, but I think is they made it much cheaper. This is another Cas protein, not from Streptococcus, but another which they have characterized. So I think that's unique in that sense.
Okay.
But there's more awareness now in India.
So there is another question from Arun. When you spoke about adaptive immune systems, the,
Ah,
the infecting virus has already put the gene in the host cell. So won't that actually affect the cell? So I think the time response kind of a,
yes.
yeah.
No, if the phage survives, because that is where the anti-restriction systems come and anti-CRISPR systems come.
Okay.
Many times, the phage survives, and if the phage survives, then that DNA is actually incorporated in the genome. Otherwise, the phage should be attacked and broken down. But once it's broken down, those pieces actually get into the host genome, and that is how it is used as an adaptive immune system. But the
Okay.
anti system, the anti restriction or the anti CRISPR are coming up in a big way again.
Okay.
Again,
So there's a question from Mother, which is on the lips of all of us, which
uh,
is some good books to read, which will help us understand the things. Uh, is there a book which addresses, touches it for the layman now?
yeah, I
Before
think there
CRISPR,
are.
the basics of, yeah.
Yeah, there are. I think just to get the idea of the principle, and it's very standard books are there. Biochemistry, molecular biology books. But if you want to read the history and all, these books are very good. Jennifer Doudna has herself written this book. I
Okay.
thought it was very nice. I haven't gone. I'm trying to get the book, but I read the preview. There are two or three books like this. I think those are the starting points to read, and
Okay.
then actually go into some. The Nobel Prize actually publishes immediately, an hour after the prize is given out, announced. They give a nice write up, about 10, 14 pages of the whole history discovery of the prize. And those are very nice. I always believe in that. I learned a lot from them.
Yeah, I have also seen that. Mother, go to the Nobel page. That's,
Foundation, yeah,
yeah, you
yeah,
should be able to get at least a starting description too. Yeah. So I don't think there are more questions, but your talk actually touched on many aspects. In addition to, of course, the specific issue of gene editing and the particular method, you
yeah,
talked about other methods, other contributors, and
yeah,
and you also talked about issues related to first authorship, which to my mind is a very ethical and important question. In fact, I recently, I mean, just yesterday, a comment by an eminent person, that Nobel Prize probably has started losing its relevance in the sense the people it is being given to are not the only contributors in many cases. So a
Yeah,
lot
I
of
know.
people work.
They certainly deserve it, but I think there are others, and
Yeah.
especially this year, I find in the internet a lot of discussion on the first authors of these papers.
Yeah, and I'm surprised that Professor Chandrasekhar, who did that sync-based
Yeah,
thing long back,
I don't.
and there is
Yeah,
a big gap between that and the cables
exactly.
and this.
Yeah,
There's
yeah,
enough time to recognize him and give him something.
I know. Yeah.
Unfortunately, it hasn't happened. Yeah. So, so it has been a really nice talk for all of us. Naveen,
Okay.
Jayanti, is there anything that I am missing? Shall we?
No,
Hello, sir.
talk.
Yeah, Sandhya, you had something to say?
Yeah, I actually have typed, two questions.
Yeah, can
Yes.
you ask? Yeah.
Yes, sir. This is Sandhya. I mean, I have a question. How do you compare the immune system of humans with that of, bacteria? Because,
Very difficult to know. I don't know. We can't compare that way. I think the thing about self and non-self is common to both the systems. Well, bacteria do it in their own way, and we do it because we are more complex, and we do it in a more complex way. Like we have the B cells, the T cells, the killer cells, and things like that. But certainly, we are evolving, right? And we have more sophisticated systems that we have, and
yeah,
probably more
that is the
efficient at things.
question. Do you think
Yeah.
human or, which is smarter? Like,
Uh, I don't know, Sandhya. This is very difficult to compare. I don't like to
okay.
compare things, but
No,
human.
because we are currently, facing with this immunity. Mostly we are talking about immunity. That is why I asked the question.
Yes, yes, yeah, yeah.
And I have one more question. Why this
Yes.
palindrome sequence is targeted? Any particular reason? Is it easy to read
No,
or
the enzymes are all, they have this diode axis of symmetry that they recognize, and therefore they act as
yeah?
dimers. They are not
Okay,
monomers, but they're dimers. There are
okay,
molecules come together to act on this. So because of this, there are enzymes which don't have this palindromic sequence requirement, and they don't act as dimers and things like that. It's an inherent feature, of the enzymes.
yeah. So
Simple.
that is how the bacteriophage escape
Yes.
the destruction.
Yeah. Exactly.
Yeah.
Yes.
Okay. 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 Thank you.
Yeah. So let me ask the audience: is there anybody who would like to ask a question?
Or they can write to me if they want.
Yeah, if you want to put it on the chat or
Right.
ask.
Right.
Yeah, it seems there are no more. In fact, so if we were face to face, you would have heard a loud applause. Because
Thank you.
this is
Thank you.
so.
Thank you.
It was extremely enlightening and interesting, and you made it so. And thank you so much from IAST Faculty Association for putting in the trouble to create all those nice slides and
Yeah.
giving
No,
such a nice talk. Thank you, sir,
thank you.
and
Thank
have a
you.
good day. Bye.
All right. Bye bye.